Okay, thanks everybody. Welcome to another edition of Conversations with Dr. Cowan and Friends. And today I have a good friend who's been here before, Dr. Mark Bailey. And the first thing I want to say is Mark just sent me the paper. I don't know if you call it a paper or dossier or something called A Farewell to Virology, which we will put a link to. And I think, Mark, maybe tell us where else you can find this. And all I want to say about that right now is I did read the whole thing yesterday, by the way, and it's, I would say, the most important paper that has been written about, well, this whole subject in the last 2 years. It's brilliantly, clearly written. You, of course, have to think a little bit to follow it, which isn't such a bad thing. It goes over everything to do with how one knows if there's a virus and how it's been not demonstrated that there is, lab leaks, all the rest of it, everything you need to know about virus in that paper. It's a real effort. I mean, this is a heroic effort for humanity, Mark. So I just want to say that up front. The other thing I want to say is there's a few things. For me, when I look at this subject, which is what I asked Mark if he could come and talk about the whole lab leak engineered virus stuff. And for me, I must say, it may be a fault of mine. When I approach this subject, I say to myself, which I'm sure you would probably agree with, Mark, if this is a chimeric or hybrid virus from a piece of HIV and otherwise a coronavirus, creating a new SARS-CoV-2 virus, sort of like if you say there's a thing called a mule, you have to prove there's a horse first and a donkey first, and then a mule. If there's no horse and there's no donkey and you can't find a mule, then none of that is true. And so I say to myself, since I know how HIV reportedly was proven to exist, which is basically a joke, because they said they found this enzyme reverse transcriptase in a culture of lymphocytes, even though the guy who actually said that, Gallo, had done a paper some years before saying that in every culture of lymphocytes, you can find reverse transcriptase. So this is, I mean, it's kind of a joke. So nobody has demonstrated HIV, nobody has demonstrated this earlier coronavirus, and nobody has demonstrated SARS-CoV-2. So I'm done there. I don't look into it any further because to me, there's nothing more to look at. There's no horse, there's no donkey, there's no mule. I don't need to know about sequences or anything. But luckily I was going to say people like Mark, but that's not actually true because I don't know any other people like Mark. So let me rephrase that. There's Mark, and he decided to look into this and explain more of the details, which I would admit I don't really have the patience to do because I already know they can't possibly be accurate. But I'm grateful that Mark has done this because I think there a little bit more needs to be said. And the only other thing I want to say is I think we would probably both agree, and maybe you could correct me if I'm wrong, that anybody from now on, once you wrote this paper or people have access to it, if they want to criticize our position or your position, they need to read that paper first and say specifically what is incorrect about it. And the reason I bring this up is because in the past, when we say things like electron microscope pictures don't prove the existence, most of the people we're dealing with actually don't know how to know there's a virus or not. You know, we're talking about people like Merrill Ness and Mercola and Kennedy and Big Tree, and they don't actually even know how you would find a virus or not. But there's a few people, in particular, Jeremy Hammond, who I must give him credit. He does actually know something about how virology is done. And it's very interesting what he'll say. He'll say, our conclusions are contradicted by our own papers that we cite. It's a very interesting comment, because I would admit he's correct. And here's what I mean by that. So, for instance, in the electron microscope paper that I refer to from, I think, the Kidney Journal, they, on the one hand, present evidence that you cannot tell the difference between a SARS-CoV-2 photo and a kidney biopsy. And yet, in their conclusion, they do acknowledge, or they do state, that they believe in viruses. So Hammond, in a sense, is correct that the conclusion of the paper is not what I came to, or I think Mark would have come to. But we're not actually looking at the conclusion of the paper, because to me, they have to say that, or at least they're choosing to say that. But what they prove or demonstrate in their paper is that you can't do that. And so their conclusion doesn't match their own data. Now, you could apply that to a lot of the papers that Mark references. Their conclusion is not that SARS-CoV-2 doesn't exist, just the opposite. They say it does. But we're not interested in that. I keep saying we as if I'm speaking for you, but I'm not interested in that. I'm interested, and I think Mark would agree, we're interested in what they actually did. And that's what we're referring to doesn't match their conclusion. So you can't have an argument, well, the paper that we cite, the conclusion is different than what we say. Of course it is. The other thing is there's this parsing of words that goes on. And let me give you an example. I said that they'd never purify the virus and use that as what they inoculate the cell culture with, right? And Hammond points out that in one of the papers, they did centrifuge the snot. And he says that is a purification step. Now, actually, he's correct. That is a type of purification, but it's pretty obvious that what I meant by that is they didn't purify it and demonstrate that they were only inoculating with a pure virus. And so just because they did a purification step to get rid of some of the debris, that's irrelevant. But that's his point. So you see, you said they didn't purify, and they did. Well, everybody knows that that's just not true, but those end up being the arguments. So I just say that because when people are thinking they're going to criticize or rebut or argue with Mark's paper, everybody should be aware that those are not real criticisms. Those are semantic tricks. So with that, a little long-winded, welcome, Mark, and tell us what you can tell us about, is there a lab-leaked virus? And thanks for joining me.
Well, thank you, Tom. It's a pleasure to be back again. And yeah, it's so strange that we're having this conversation because realistically, the laws of biology and nature don't change based on stories that people make up. And really, when we're looking at things like lab leaks and gain of function, unfortunately, people seem to think that there's a product coming out when we're saying to them, well, there's no product going in, so there's no product coming out the other end.
Yeah. And
the problem is that I think the excitement of the kind of, it's like a movie plot line. You know, you think back to the 1990s movie Outbreak, and the bad guy played by Donald Sutherland, I think, played the bad guy, and he's getting the sample of the virus, you know, the virus, and he's putting it in a vial, and he wants it for military purposes to take away. But obviously for you and I, we just look at it and say, well, what's in the vial? Because we can't see evidence of a virus in the vial.
Right.
So there's nothing going in, so there's nothing going out. But anyway, I thought what we could do is the article you mentioned recently, which appeared on the Children's Health Defence site and written by Joe McCullough, is actually a really good article to look at, because it kind of deals with all the fallacies that we need to talk about here. So I will now, just to let the audience know, Tom and I are not the tech guys, and but we should be able to do a screen share.
That's an understatement.
Yeah. I'm like you, Tom. I don't have a smartphone. I've still got a 3G flip phone.
Yeah, I don't even have that.
But let's see. I'll attempt to screen share here, and here we go. So can you see that? Okay.
Yeah, I got it.
See that? Good. All right. So yeah, this was the piece that you've talked about recently in your podcast that appeared in the Defender. Fauci's team involved in research to create deadly version of Spanish flu virus.
Right.
So essentially, it sounds very spectacular, but what we need to do is just break down what they're actually talking about. And first of all, I mean, many of your audience will be aware that there was no evidence of a Spanish flu virus. There was certainly evidence of people dying in 1918, but all of the clinical experiments that were carried out around that time, famously the Rosenauer experiments,
right,
could not demonstrate that there was a transmissible agent. And they tried everything. I mean, we're talking about coughing in people's faces, taking snot and putting it up their nose and injecting it into them. Yeah. And absolutely nothing was shown to be transmitting. So that's the first point we should take into account. So when they're talking about deadly version of Spanish flu virus, we had no evidence whatsoever of a virus. So getting down, so now, Thomas Renz.
Do you know, by the way, Mark, do you know if anybody even did the sort of bogus, quote, isolation experiments with what they call the Spanish flu virus?
Well, I mean, obviously at the time they didn't have.
Yeah, they didn't, because it was almost 30 years before they even had any way of doing an incorrect isolation experiment.
Yeah, well, I mean, at the time, they didn't know what was going on. So they were just trying all sorts of things. I mean, they thought it was a bacillus. So they were trying to isolate that with their usual kind of bacterial cell culture techniques. But yeah, I mean, in short, no. And we'll get into that soon about what they actually think that they're resurrecting. So yeah, and then so Joe McCullough refers to a report by the attorney, Thomas Renz, and I don't know too much about Thomas. He seems to be a genuine guy, but like you were saying earlier, maybe someone that doesn't really understand the virological science. So Thomas Renz reported that these mad scientists are testing recreated Spanish flu on monkeys. All right. So, but if we actually look at the papers that they're talking about, first of all, I'll try and bring that up. Okay, so this was the paper that got them all concerned. Pandemic 1918 influenza virus does not cause lethal infections in rhesus or homologous macaques. Now, the problem with this paper, again, is what we're talking about earlier, is what they were actually testing here. And the input agent was a culture that was prepared with, it was maiden Derby canine kidney cells, I believe, for this one. And they took some complementary DNA plasmids and introduced them. They did a transfection experiment with the canine kidney cells. And then out of that, they said they had a virus. Now, of course, the reason they're claiming that is because they've gone to GenBank and said that the sequences they were inserting, transfecting, came from the alleged influenza virus. But we know that's not correct because they've never shown the influenza virus to exist, and they've certainly never shown these sequences to come out of a particle that fulfills the description of a virus. So essentially, that's what they're starting with, a canine kidney cell soup with various genetic sequences in it. All right, and so that was the starting
So they
point.
had a gene bank sequence that they got, who knows how, from what, and they said they attempted to put that into the culture.
Yeah, exactly. And I talk about this extensively in the paper that I've just published, that they don't establish the provenance of these sequences. They just, unfortunately, they claim that they're viral. And then once they've done that, forevermore, anyone else detecting the sequences also says that they've detected the same virus or the same viral sequences.
Right.
But obviously they don't have that anyway. So in this study, first of all, they talk about the mouse lethal dose. Now, this is just a joke because nothing killed the mice apart from the euthanasia that was given to the mice. So they essentially said that they infected, in quotes, the mice, and then once they lost 25% of their weight, they killed them and said that that represented they could estimate the lethal dose of the virus. So obviously, I mean, that's a complete joke. And then, so what did I want to talk about next? So this experiment they did with the monkeys is again one of their animal study kind of joke studies, where what they do is that they say they're infecting the monkeys by pouring a biological soup directly into their lungs. So here we've got an intratracheal dose. So that's pouring it directly into their windpipe.
And it is this kidney culture with a transfected sequence in it, or a canine culture.
Yeah, the canine kidney cell culture.
Canine kidney cell.
It's just, as you know, Tom, this is just a biological soup. It's not purified particles in any way.
Yeah.
So they pour it straight into the monkey's lungs. Now, this is, I mean, you and I would not call this, I mean, both of us have problems with the concept of infection coming from a terrain point of view, but this is not anything like what would happen in nature. Pouring, in this case, I think they put about 4 mils of fluid directly into the monkey's lungs.
Yeah.
Now, these are little monkeys. I mean, some of them are only about 4 kilograms. So for us, that would be like equivalent to pouring a quarter of a cup or a third of a cup of biological muck directly into someone's lungs, which is, of course, that doesn't represent anything like what would happen in nature.
Right.
And as you know, even getting any fluid into your lungs, I mean, if people think about when they swallow something, a drink, and it goes down the wrong way, I mean, that's just a tiny little amount of fluid going in, and you know the violent reaction you get to cough it back up again. I mean, your lungs don't want this fluid being poured into them. So this is a stupid method of exposure, which doesn't replicate anything like what would happen in nature. And not only that, they did a second experiment where they went further than the trachea and poured the fluid directly into the left bronchus. So this is incredibly invasive now. Now we're getting deep down into the lungs and pouring fluid right into the lung tissue itself. I mean, so absolutely stupid. And not only that, but then as part of their experimental process, they were taking samples every second day, doing bronchoalveolar lavages. I think we've got the diagram down here. And you can see here the BAL being performed every second day on these. Wow. So, I mean, again, they are anesthetizing the monkeys. They are putting a scope, and don't know about you, Tom, but I sat in on one bronchoscopy clinic once.
Yeah.
And it's a pretty traumatic procedure.
No kidding.
Yeah. that horrible tube going down your windpipe and into your lungs. And they were doing that every second day to the monkeys. And then they flush in a whole lot of fluid
Yeah.
and then suck it back out again. And with these poor little monkeys, they were doing that multiple times every second day. So we just need to explain this to people so they can see what actually goes on with these monkey experiments. Because, I mean, you and I think if we were designing the experiment and we thought there was contagion, we would just get our sample and aerosolize it into the cage, and they could just breathe it in. And surely, if it's a highly infectious agent, they would all just catch it that way, so to speak.
Yeah, I would purify the virus first and put that in some saline, and then do a control of just saline and spray it in the cage. And I wouldn't actually keep a monkey in a cage, but I know what you mean.
Exactly, yeah, and we're not advocating this sort of thing. We're just talking about what you do for a scientific procedure. Or
Right.
even better, Tom, you just have one monkey who's got the flu and then just pop them in the cage with the other monkeys, and they should all get it, shouldn't they, within a couple of days? But
yeah, you know, it's interesting you say that, because I would even want to control for the cage, put the monkeys in an African game park and see what happens.
Totally, yeah. So we're talking, I mean, these experiments
I put animals in cages, and I can tell you for sure they don't like it.
No, and that's another thing. Yeah, it's a traumatic experience.
Yeah, this is animal abuse.
It is, yeah, and that's in my paper. That's what I called it, animal abuse studies.
Yeah.
So, and of course, Tom, there's no control study here. So they're not taking a similar group of monkeys and pouring biological soup said not to contain the virus directly into their lungs, and then traumatizing them for the next couple of weeks with all these procedures. So, occasionally you'll see control studies, but they're very disingenuous. Like they'll involve phosphate buffered saline or something much more innocuous than these biological soups that they're pouring into the monkeys.
Right.
So anyway, well, what happened here? Well, the monkeys didn't really get that sick. They were all pretty much okay, despite this.
Hopefully, they bit the lab people, but otherwise,
hopefully, yeah, they made some complaints, but about their treatment when they were staying there. But yeah, we can see that didn't really do anything, and this often happens, is that clinically they don't even notice anything. The monkeys don't really exhibit any symptoms and signs, despite the fact they've allegedly had massive doses of viruses poured directly into their bodies. Now, of course, after this, they said they confirmed, in quotes, that there was a virus present, because they did the old cytopathic effect culturing, and again, didn't perform any valid controls. And they did a whole lot of blood tests and stuff, and said, wow, we found all these inflammatory markers in their bloods, which tells us that they were infected with a virus, which, as you know, is complete nonsense. They can't make that sort of claim based on such indirect kind of evidence. So anyway, the conclusion.
And Mark, isn't it true that sometimes they'll put some sequences in, and then they test for the presence of the sequence, and they find the same sequence that they put in, and they claim that proves they had a virus?
Completely. And again, it's totally irrelevant. Yeah. And Tom, the thing is, they only ever find the sequences in the same place that they've put them.
Yeah.
So there's no evidence that the agent that they've put in has any invasive characteristics. So if they put them in the lungs, they find them in the lungs, if they make them swallow them, sometimes they find them in the feces, all this kind of stuff. But there's no indication whatsoever that they're dealing with an invasive agent. So anyway, the conclusion of this paper was that because this alleged virus didn't seem to do anything, they may need to engineer it a bit more to make it more pathogenic, which has upset people like Thomas and Joe McCullough, etc., who think that this is just a terrible idea. So, I think that.
So the real conclusion was there's no evidence of a Spanish flu virus, number one. And well, that's sort of all you need to say, except that what they call the virus and what they call sort of upgrading it actually doesn't do anything.
No, there's nothing to upgrade. So, I mean,
Yeah, right.
as you know, you can make toxic biological soups, but it has nothing to do with viral particles. If you don't show there's a viral particle in there, then that's not the problem. So,
Yeah.
yeah.
Right.
So that was the first one, and then we get to the next part of the article, which says that there's been a cover-up of the SARS-CoV-2 origins, apparently. Now, this is a little bit amusing because I'll see if I can bring up the other article here. Just bear with me. I'll just get that one. Right, here we go. Okay, so can you see that okay?
Yep, got it.
Yeah, so this is April 2020. And interestingly, this whole lab leak story was being promoted as early as February 2020.
Yep.
And Eric Coppolino's actually got the COVID chronology, which has the timeline, and it's really useful for checking all this stuff. So for those people that think the whole lab leak thing has been covered up, I mean, it's not really consistent with the mainstream media coverage that we're seeing. Now, this photo first appeared in 2018, and it came from the China Daily, which is a state-controlled media agency. So this is the communist Chinese government releasing these pictures in 2018. So I don't know about you, but that doesn't sound like me, like they're trying to keep it all top secret when they're claiming that there's all these deadly strains of viruses, including coronaviruses, in this fridge that they've got. That seems
which
to
they
me
forgot to defrost or something?
Yeah, and they were concerned that the seals weren't too good around here.
Yeah.
And that the deadly virus might have been leaking. Now, keep in mind, Tom, look at the date, April 2020. Now this, if I'm correct, this is when Andy Kaufman is making videos saying that there's no virus, and they are being struck down within hours off the internet, you know, and I think David Icke similarly, when he was saying there was no virus. So all those guys that came out saying, look, there's no virus, you better have a look at the science here, they were being completely shut down while this sort of story was being splashed all over the mainstream media.
Yeah.
And was freely circulating on Facebook, etc. And not only that, we had the US Senate openly talking about this stuff in mid-2021. They were doing press conferences saying, look, we're concerned that this thing leaked out from a lab, etc. So again, I think the people who think that there's some sort of cover-up going on here, I would say just go back and have a look at 2020 and see all the stories that were appearing all over the mainstream media saying that they were engineering viruses. The whole Wuhan Institute of Virology that was being splashed around everywhere in January, February 2020 onwards. So to me, this was all intentional placement. It's nothing to do with cover-ups. Cool. So on to the next one I wanted to talk about. So lack of transparency breeds mistrust. And now we get to the Jeffrey Sachs opinion piece that was published a couple of months back. And you'll be familiar with that one, the one he co-authored.
Yes.
Yeah.
And by the way, I don't know if you knew this, but Jeffrey Sachs was my mother's bridge partner when I was growing up and my sister's best friend.
Yeah, you really got to talk to him about the state of the virological science because, I mean, certainly the problem with Sachs and Neil Harrison is that they take all of virology's claims at face value, and that's where they start. So when they produced their opinion piece, they didn't do an analysis of whether the science was correct or not. They just said, yes, all this stuff's going on, there's gain of function research, etc. It's all happening. So one of the big things that Sachs and Harrison were concerned about was the supposed research being done by the likes of the Eco Health Alliance, Peter Daszak and co, and Ralph Barrick at the University of North Carolina, Chapel Hill. And realistically, if you actually look at the papers that they published, once again, they're a complete joke because they don't show any viruses. What they show is, I'll give you an example. There was one paper where they went to bat caves in China and they collected a few hundred bats and they did PCR tests and quotes on them and said that, hey, these bats are infected with coronavirus. Now, the bats are 100% well. They're not exhibiting any symptoms or anything. So obviously, they're not detecting viruses. They're just detecting sequences said to come from viruses. And then they dissected the bats and ground up their intestines. And first of all, they tried to do the usual culturing with cytopathic effect methodology, but that didn't work, unfortunately for them. They couldn't even use that fraudulent technique to show that they had viruses. So they went to their next stage of research to show what they claim was pathogenicity, and they used the ground up bats' intestines and injected it straight into the brains of baby rats. They were just a couple of days old, and amazingly, it didn't kill. A lot of them survived this kind of ridiculous sort of trauma. Right. But then they euthanized the rats and cut open their brains. And they saw, some vesicles inside the brain. And then they said, well, that's it. That's the virus.
That's the proof of infection.
Proof of infection, Tom, is apparently by taking biological muck and injecting it directly into the brain of another animal and then seeing a couple of vesicles here and there and claiming that must be the virus. And I mean, it's a total joke, as we were talking about with electron microscopy, is that it's totally out of context. All they have is some brain tissue with a couple of vesicles with an arrow saying, well, that's the virus right there. Yeah. And then, of course, they confirm, in quotes, that it is a virus by performing the same PCR protocol that they did at the start of the project. And then they say, well, look at that. We detected the very same sequences in the brain that we just injected into the brain a couple of days earlier. So we're not showing anything. It's a total joke. And I think we should also point out, this is where the whole zoonotic nonsense comes from. This claim that all these pathogens are sitting in bat caves and swamps, ready to jump out at humanity.
Yeah.
And even on their own terms, it makes no sense because, as you know, when they were first describing coronaviruses in humans, or what they said were coronaviruses in the 1960s, well, post-World War II, in the Common Cold Unit, they were telling us that, these are the joke viruses that just cause a runny nose, and that's about it, basically. They don't really do anything else. And now we're supposed to believe that they're incredibly deadly pathogens that can absolutely wipe out humanity. So, even on their own terms, the stuff doesn't make any sense. But anyway, that kind of sums up that. I think we've probably covered that the whole Chinese bat cave gain of function coronavirus stuff is a complete joke. And in fact,
and you went over in your paper the whole history of coronaviruses, which is basically finding a sequence, compare it to a previously similarly found sequence, compare it to a similarly found sequence, and that one was made up.
Well, totally. So I went back, I just followed the trail and just kept following the citations to see where they went back to, and saw, because as you said, people like Jeremy Hammond, he's claiming that these sequences are specific to coronaviruses. And therefore, if you find the sequences, it means that you've got the smoking gun, that you've found the coronavirus. So, but anyway, I went back to the initial descriptions of the first alleged coronavirus genomes, which was in the 1980s. And you get to papers which claim that they'd purified virions, but there's nowhere in those papers that they demonstrate that they have viruses. They just assumed that they had viruses, and like you say, do a bit of centrifugation and detect some sequences, and then say, hey, presto, we've just isolated the viral sequences. When the whole key, and I think you were pointing this out at the start, we're not interested in what they claim at the end of the paper. We're looking at the methodology.
Yeah.
And we're saying there's no way you can make those claims from the methodology that's been employed. It's simply invalid. And the other thing we should also point out, which I cover a lot in the paper, is there's no control experiments. So why not just do it? So with the original coronavirus genomes, they said that they were culturing these in their brews, but there was no control experiment to say, well, let's just do the same culturing technique, but we won't add the quote virus this time, and see what sequences that we can get out of it. But in the history of virology, they've simply avoided this, and we know why, because it would just unravel the entire thing.
Yeah, right. And again, it's not the conclusion that we're using these papers. We're showing that virology disproves itself.
That's right. Yeah, I mean, that's essentially what we do when we do an analysis of it. We just break down the papers and the methodology and say, well, that's not a controlled experiment, so that can't be presented as scientific proof. Or we just point out that they don't have direct evidence, so they can't find the particles in the organism for a start. So that's a major problem for their hypothesis anyway.
Yeah.
So yeah, it all falls apart. Now, so the other thing was too, is that people were very impressed with another alleged leaked document. And this relates to this whole suspicious coincidences abound. Now, actually, Sam's got a video. We did a couple of videos on gain of function. One was gain of function gaslighting, and one was gain of function garbage. And I think in that second one, we covered the diffuse proposals, supposedly, that were, that the public's found out about through these leaked documents. Now, again, I find it rather strange that all of these documents are in the public arena, and yet we're told they're top secret. And it just seems, again, a little bit strange. Yeah. I'm sure if they had secrets they wanted to keep, they'd be keeping them. Yeah. But anyway, I took the time to read through all of those grant proposals related to defuse, and that was submitted to DARPA. And officially they said they didn't get approved, but Sachs and Co were saying, well, maybe they carried out these gain of function experiments anyway. Now, the thing is, again, if you read through these papers and these submissions, there's nowhere in there that proves that there are viruses. They simply just talk about it. Yeah. And, they'll talk about a SARS-CoV backbone that they got from bats, and then they're going to engineer certain bits like a new furin cleavage site and all this kind of stuff. But if you actually look through the papers, there's nothing there which proves that they can do this. So again, it's just complete smoke or mirrors. And I think, unfortunately, what happens is that people see these sorts of headlines and stories like defuse and think that it's the smoking gun, which unfortunately these guys seem to do here.
Right.
All right. So moving on, there was another claim. I don't know if it was in this article. Oh, no, it was. Yep. So patents prove SARS-CoV-2 is a manufactured virus. And this related to, I think this was a mid-2021 paper, article written by Joseph McCullough again. And again, I don't know about you, Tom, but I don't think patents prove anything. Like you and I could... You and I could patent a magic flying carpet, but as long as we're the ones that come up with the proposed idea and we're the first ones to do it, we can take out that patent. And as you know, a patent doesn't mean that you have made the product or that the product is proved to exist. It simply means it's like an intellectual property right. It really isn't. Right. You're the first one to go in and say, look, if this is ever done or created, I want to have the complete rights to it, etc. So, I mean, we can see the fallacies that a patent could possibly prove that a virus exists. So once again, you have to take
Mark, that's why I like this question, that how does a virologist prove the existence of a virus and show it causes disease? So according to this, the answer would be, so a virologist, man or woman, shows up to work, puts on his hazmat suit, sits around the table with 10 other virologists and says, hey, does anybody write a patent this weekend? Somebody said, yeah, I did. Here it is. This is a guy that got a patent right here. He said, there you go. We got it. We proved the virus. Because when you put it like that, most people would say, well, of course, that's not what they do, because it's ridiculous. Yet they'll show us the patent proving that the virus exists, as if that is exactly what they did. They made a patent, so it got to be true.
Totally. And Tom, the thing is that when you look up these patents, they refer to exactly the same studies that we've dissected many times over in our papers and presentations, etc. So we're not seeing anything new here. And people think that they're finding smoking guns, etc. Now, the thing is, where it gets confusing is that a lot of the patents relate to things like the spike protein. Now, you and I are not saying that there are not proteins in these cell culture soups, and some of them they call spike proteins. Well, that's fine if they want to do that, but what we're saying is that there's no evidence that these proteins that they have in their soups come from an infectious replication competent particle, i.e. a virus. So we don't have a problem that they're claiming to be fiddling around with various proteins and sequences. The issue is that you need to have the whole package, which is the virus,
And I think you would probably agree, we're not even necessarily saying they're not trying to fiddle with proteins to make things that might injure people. They may be. That's not what we mean by a virus, though.
Totally. And I think the first Pfizer actually took out a patent on a spike protein, probably for the purposes of producing what they said was a vaccine, like two decades ago. So this stuff has been in the pipeline for a long time. But a lot of it, it relates to injectable products, not to viruses. So now the other thing related to this is David Martin's dossier that he published also in 2021. Now, again, people thought this was a smoking gun, and I think it was a little bit confusing for people because now I don't know, have you spoken with David Martin before?
Yes, I know David.
So you know David, so that's great. I've not had the privilege of speaking to him before, but I've seen several of his presentations and I've read his dossier. But one thing that he makes clear at the start of the dossier, which I think most people miss, he says throughout this document, uses of terms commonly accepted in medical and scientific literature do not imply acceptance or rejection of the dogma that they represent. And I think that's probably one of the most important sentences in his entire dossier, that David is not claiming that what is reported in these various federal grants and patents are, consistent with biological plausibility or biological reality. And I think that's what a lot of people have missed. That they think what he's saying is that everything in this document involves the alleged gain of function research, etc., going on when he hasn't made that claim at all. And I think one thing that I'm not 100% sure about, I don't know what David Martin's position is with the existence of the virus. Sometimes he is very much seems to be in the no virus camp, and other times he seems to suggest that perhaps there are viruses. I'm not sure on that. I believe I don't know what you know about this, Tom, but I don't think he thinks SARS-CoV-2 exists, but I could be wrong on that.
He's all over the map.
Okay. Yeah.
Depends, I think, on who he's talking to.
Okay. Yeah. Yeah. Well, in any case, I think it was a mistake that people thought that this was some sort of smoking gun. And I mean, the other thing was that people who think that Fauci is going to get in trouble for his activities in the last 2 years, I think is just a complete joke, because as you know, he's been on the scene since the HIV/AIDS scam, and he's been through all of these things many times. And there's been so many scandals for the 4 odd decades that he's been running the show that I don't think he'd be too worried about any of these latest allegations against him. So,
Yeah.
yeah.
You know, it reminds me of, when Trump was running against Clinton, every rally they had before the election, there was the chant of lock her up, lock her up. And he always said that. And that, I think, probably helped him win, if in fact he did win. I don't know that. But
Yeah.
then I think it was the day after he was inaugurated, he said, never mind, it's too divisive. We don't want to lock her up. And if I was somebody who that's why I voted for him, I would say, wait a minute, that's why I voted for you. And they keep doing that same thing. Oh, we're going to get this guy. We're going to get this guy. And then never mind.
Totally. Yep. I think Fauci
it's just playing people for suckers.
Yeah. Nothing's going to happen to Fauci. He's going to enjoy his retirement by all accounts. So yeah, I think it's a waste of time. And as we know, I mean, going after these individuals, it's not the problem's not the individuals.
It's not the individual. They get a new one. A new Fauci worse comes next.
Totally. Yeah. So another thing we should talk about too is, you mentioned the whole HIV sequences thing. And, this is another problem, because what they want us to do is accept that there are such a thing as HIV sequences.
Right.
And we know there are not, because it's the same problem. They can't show that the particle exists. And I mean, this debate was going on back in the 1990s. And this is actually quite a good paper. It's quite technical, but Eleni Iliopoulos and the Perth group wrote this paper. I think it was in Continuum, Why No Whole Virus. And we're responding to Peter Duesberg's claims that these sequences were specific to the virus. And if you like, it's a more detailed and more technical version of the sort of stuff that Jeremy Hammond brings up, trying to say that these sequences come from viruses. And Eleni basically lays it out and says that they're not viral sequences because you don't have a virus, is the summary of the paper. So this claim that SARS-CoV-2 has HIV sequences, mixed in with it is just as stupid, because when people say HIV sequences, what they do is they look up GenBank and go, oh, here you go. This is proof that HIV exists and these are the sequences. But once again, you and I are not disputing the fact that they can do experiments and purport to detect these sequences. We're just saying that they don't come from a virus. They come from somewhere else. So
right. And therefore you cannot get a replication competent protein coded genetic material inside infective particle when there was never part of the experiment.
Totally. And I think I can't remember it was an Alex Jones show, I think last year sometime. And he said, they've created this chimeric monster with HIV and SARS-CoV-2 and some other virus. And, he put up this paper on the screen, I think. And I thought, well, I'll read the paper. And the paper had no virus in it, of course.
Yeah.
All they had was various sequences that they'd thrown together, which they claim belong to different viruses. And they'd created a very long strand of nucleic acid and said, wow, look at this, we've created this super virus. But it didn't do anything. It was just a whole lot of sequences
on a
thrown
computer.
Together.
Yeah.
So what is the danger because of all this stuff, because there's been criticism towards those of us in the no virus camp that this is a distraction, that there's all these globalist plans and new world order coming into play, and we need to just focus on that rather than whether viruses exist or not. And I would just say that I haven't had a chance to read this entire report because it's pretty long and it's just come out a couple of days ago, but this is another one headed by Jeffrey Sachs, and basically giving a rundown over what they say happened over the last 2 or 3 years with the alleged COVID pandemic. And of course, once again, it comes into the whole possibility that this is a virus that leaked from a lab. So they're bringing up that story once again. And if you actually look at where this leads, the recommendations all lead back to the stuff that we are warning against, which is the biosecurity state, Agenda 2030, sustainable development goals, all this technocratic globalist rubbish that they're trying to bring out. And they are using the virus narrative to help bring in this new world order. So there'll be massive amounts of restrictions. This could be all sorts of things that you and I have talked about before, like restricting travel, restricting ownership of your own animals, restricting where you can get food from, etc., all on the basis that there are these incredibly dangerous pathogens that are about to spring out. So
yeah, in other words, I think, and tell me whether you agree with this, I think the gambit here is, okay, there's this lab leaked virus, therefore science experiments, particularly around virology or microbiology, are dangerous. Therefore, so that's the problem. So therefore, all science, or at least all, microbiology experiments, need to come under a responsible organization, let's say the World Health Organization. So if you want to do anything with animals or microbes, you have to clear it through the World Health Organization. So we're the arbiters of what is called science. And so, then you have this situation where science becomes under the umbrella of a central body, because obviously you can't trust these people to do it by themselves.
Totally. And Tom, if we look at the start of the report here, I mean, it's just terrible. It says that, too many people, often influenced by misinformation, have disrespected and protested against basic public health precautions, and the world's major powers have failed to collaborate to control the pandemic.
Right.
Now, that should strike fear into anyone who values freedom, because what they're basically saying is that the likes of the Lancet Commission and the WHO and the UN are the ones that should decide what is misinformation. And they're saying that we need public health precautions, which ultimately is based on what? Is based on this bogus concept of contagion. That's how they're bringing the whole thing in. If people know that contagion, pathogenic viruses don't exist, then they ignore all of this stuff.
Yeah,
right
this
from the
all
start.
goes away.
Yeah, because I don't know. I mean, this too. Back in 2020, and your family was probably like the Baileys, when they said that they were going to lock things down in March 2020. We were one of the only people going, no way, this is ridiculous.
Ridiculous.
And the problem was that even people who valued freedom but believed in contagion were the ones that actually went along with it. And they stopped going along with it when it got too bad, or when they were forced to be injected with toxic biologicals. And, it was, I think those of us who don't believe in contagion were the ones who could see right from the start where this was heading. And that's why we're encouraging more and more people to see through this, not only the nonsense of lab leaks and engineering pathogens and all this kind of stuff, but just to see that there's no contagion. And like we're happy if someone sends us a paper which shows contagion between animals or between humans, we're happy to look at it. But if it involves pouring biological soup directly into animals' lungs and then claiming that that's contagion, we're not buying it for a second.
Yeah. All right, let's finish with, and let me lay out the challenge, and I'll see if you agree that here's what we're asking people like Mercola and Mr. Kennedy and Professor Sachs and David Martin and Tom Rentz and Meryl Nass and a whole lot of other people, all the people who believe in this lab engineered virus. As far as I can see, at this point, you have two options. One is show us the proof that this lab leaked virus actually is real. The way to do that is, number one, demonstrate that HIV is real and exists. Number two, that the previous coronaviruses are real and exist. Number three, there is this SARS-CoV-2 that you can find in some sick person that you say has COVID. And then show that these sequences that you're saying were mixed together actually came from those particles. So if you can do that, we will, at least me, I will admit that I was wrong and that there is a lab leak virus. Show me the HIV exists, coronavirus exists, SARS-CoV-2, and all the sequences that you allege came from them do in fact came from them, and then maybe show us how they moved the sequence from HIV into the new coronavirus. If you can't do that, I would say your other option is to publicly admit that you're incorrect. Because if you don't, now that you know that there is no scientific basis for this lab leak theory, you become an accomplice in this crime of locking the world down even further with more central control when you knew, as of now, that it was simply based on a lie. So I don't know if you would go along with that, Mark, but that's how I see it.
Absolutely, Tom. No, I agree with that. And I'd also encourage people to read the paper that I've just put out called A Farewell to Virology, Expert Edition. Now, I think many of your audience will be experts and they will be able to have a one-stop shop to see that there's just no science behind this stuff. And as you say, if we've missed something, if we've missed a crucial paper or experiment, we would be more than happy to have a look at it. But from what we can see, you know, there's just no basis to this whole thing. So unfortunately, I think it all comes back to this fear narrative, and that's what we're trying to get people out of. There's no need to be scared of contagion or being scared of being around other people. And unfortunately, the whole lab leak engineered virus stuff brings people straight back into that fear narrative. And it really is, it's unhelpful, and people will eventually manifest with, you know, not only psychological distress, but potentially physical illness as well.
Yeah. Okay, Mark, I think we got it. I always appreciate everything you're doing. It's just a real gift to humanity. I don't know what else to say about it. So that's how I see it. And we'll link to your article. Everybody who's interested in this, I think it's a must read. Like I say, it's the best written thing that I've seen about this whole virology thing, I think, ever. So there you go. Mark, thanks for joining me, and we will be in touch.
Thank you, Tom.
Okay, take care, Mark.